Electrochemical sensors, principles, classification, materials, and applications. Electrochemical sensors represent one of the most significant classes of analytical devices in modern chemical, biological, and environmental monitoring. According to the International Union of Pure and Applied Chemistry, IUPAC, a chemical sensor is defined as a device that converts chemical information, ranging from the concentration of a single analyte to a full compositional analysis, into a measurable and analytically useful signal. Fundamentally, such sensors consist of two key functional components: a recognition element, receptor, and a physiochemical transducer. The receptor is responsible for selectively interacting with the target analyte and may include functionalized surfaces, ion-selective membranes, enzymes, antibodies, nucleic acids, or synthetic molecular structures. The transducer converts this chemical interaction into a quantifiable electrical signal. Among all chemical sensing technologies, electrochemical sensors have emerged as dominant due to their high sensitivity, low detection limits, often reaching picomolar levels, rapid response times, and relatively low instrumentation costs. These sensors are highly versatile and exist in configurations ranging from conventional laboratory-based systems to miniaturized, portable, and wearable devices. Their primary function is to provide accurate, real-time information about the chemical composition of a system without significantly perturbing the sample. In practice, electrochemical sensors operate by measuring electrical signals generated from redox or interfacial processes occurring at the electrode surface upon interaction with the analyte. Classifications. Electrochemical sensors are broadly classified based on the nature of the measured signal. The principal categories include potentiometric, amperometric, impedometric, or conductometric, photoelectrochemical, and electro-generated chemiluminescence sensors. Each type relies on distinct physiochemical principles governing the interaction between the analyte and the electrode interface. Potentiometric sensors operate under near-zero current conditions and measure the potential difference between a working electrode and a reference electrode. This potential arises from the establishment of a local equilibrium at the electrode interface and is related logarithmically to the analyte concentration through the Nernst equation. These sensors are among the oldest and most widely used, with applications dating back to the early 20th century. Ion-selective electrodes, ISEs, coated wire electrodes, and field effect transistor-based sensors constitute the primary subcategories. The selectivity of these devices is largely governed by the composition of the sensing membrane, which incorporates ionophores or dopants tailored for specific analytes. Glass electrodes, particularly for pH measurement, are the most ubiquitous example. In addition to hydrogen ion detection, specialized glass compositions enable the measurement of alkali and alkaline earth metal ions such as sodium, potassium, lithium, and ammonium. While highly effective in aqueous systems, these electrodes require careful calibration in non-aqueous environments. Amperometric sensors, in contrast, function by applying a controlled potential to drive electrochemical oxidation or reduction reactions at the electrode surface. The resulting current is directly proportional to the concentration of the electroactive species. These systems typically employ 2 or 3 electrode configurations, consisting of a working electrode, a reference electrode, commonly silver, silver chloride or calomel, and a counter-electrode made of inert materials such as platinum or carbon. Supporting electrolytes are used to maintain ionic strength and minimize solution resistance. The theoretical basis for amperometric sensing is often described by diffusion-controlled processes, where current-time relationships follow established electrochemical models. Due to their high sensitivity and fast response, amperometric sensors are widely used in clinical diagnostics, environmental monitoring, and industrial process control. Impedometric sensors, also referred to as conductometric sensors, measure changes in the electrical impedance of the electrode interface upon analyte interaction. These measurements are typically performed by applying a small amplitude alternating potential and analyzing the system response over a range of frequencies. This technique offers several advantages, including high sensitivity, the ability to probe interfacial processes, and compatibility with steady-state measurements. Conducting polymers such as polypyrrole and polyaniline are frequently employed in these sensors due to their tunable electrical properties and ability to interact with specific analytes, including gases like ammonia and volatile amines. Applications of electrochemical sensors. The application scope of electrochemical sensors spans environmental, biomedical, industrial, and pharmaceutical domains. Their popularity is driven by inherent advantages such as low detection limits, rapid response, operational simplicity, and compatibility with miniaturization. Advances in nanotechnology have further enhanced sensor performance by enabling the integration of nanomaterials such as metal nanoparticles, carbon nanotubes, graphene, and metal organic frameworks. These materials provide high surface area, improved electron transfer kinetics, and enhanced catalytic activity, which collectively increase sensitivity and selectivity. Biosensors represent a specialized subclass of electrochemical sensors that incorporate biological recognition elements. These include enzymes, antibodies, nucleic acids, and whole cells, which interact selectively with target analytes. The resulting biochemical interaction is transduced into an electrical signal and processed into a readable output. Electrochemical biosensors offer distinct advantages, including rapid analysis, minimal sample preparation, and high specificity. They are widely used in medical diagnostics, particularly for glucose monitoring in diabetic patients, where enzyme-based sensors employing glucose oxidase are standard. The detection of biomolecules such as hormones, enzymes, and nucleic acids is of critical importance in clinical diagnostics and biomedical research. Traditional analytical techniques, including polymerase chain reaction, gel electrophoresis, and immunoassays, provide high accuracy but are often time-consuming, labor-intensive, and require sophisticated instrumentation. Electrochemical methods offer a compelling alternative due to their speed, cost-effectiveness, and potential for miniaturization. Integrated biosensor platforms have been developed for detecting biomarkers in biological fluids, enabling early diagnosis and improved disease management. Electrochemical biosensors have also demonstrated significant potential in the detection of viral infections. Techniques based on DNA and RNA electrochemistry have been employed for identifying pathogens such as influenza, HIV, hepatitis viruses, and emerging infectious agents. Aptamer-based sensors, which utilize short oligonucleotide sequences selected through in vitro evolution processes, offer high specificity and sensitivity. These APTA sensors detect target molecules by monitoring changes in current or impedance resulting from binding events at the electrode surface. Such approaches have gained attention for their application in rapid and cost-effective diagnostic tools, particularly in point-of-care settings. The global health crisis caused by viral outbreaks has further underscored the importance of rapid diagnostic technologies. Electrochemical sensors have been explored as complementary tools to conventional molecular diagnostics, offering faster response times and lower operational costs. While not intended to replace established methods entirely, they provide valuable support for large-scale screening and monitoring applications. Enzyme-based electrochemical sensors constitute another important category, leveraging the catalytic activity of enzymes to generate measurable signals. Enzymes accelerate biochemical reactions with high specificity and efficiency, often increasing reaction rates by several orders of magnitude. Immobilization of enzymes onto electrode surfaces enables the selective detection of substrates, with signal generation mediated by direct electron transfer or through redox mediators. The performance of such sensors depends on factors including enzyme stability, immobilization technique, and electrode material. Advances in nanomaterials have significantly improved enzyme loading and electron transfer efficiency, enhancing overall sensor performance. Ion-selective electrodes continue to play a central role in clinical chemistry, particularly in the measurement of electrolytes in physiological fluids. These sensors are routinely used for determining concentrations of ions such as sodium, potassium, calcium, chloride, and magnesium in blood, urine, and other biological samples. Their ability to measure ion activity rather than concentration provides clinically relevant information related to physiological conditions. Despite challenges associated with in vivo applications, including biofouling and material stability, ongoing developments in microfabrication and biocompatible materials are expanding their potential for continuous monitoring. Environmental monitoring represents another critical application area for electrochemical sensors. The detection of trace levels of pollutants, including heavy metals, organic contaminants, and inorganic ions, is essential for assessing environmental quality and ensuring public safety. Electrochemical stripping analysis is a powerful technique for trace metal detection, involving a pre-concentration step followed by electrochemical measurement. This method offers exceptional sensitivity due to the accumulation of analytes at the electrode surface. Advances in sensor miniaturization and portable instrumentation have enabled on-site and real-time environmental analysis, supporting decentralized monitoring strategies. Nanomaterials in electrochemical sensors. Nanomaterials have revolutionized the design and performance of electrochemical sensors. Carbon-based materials, including carbon nanotubes and graphene, are particularly significant due to their unique electrical, mechanical, and chemical properties. Carbon nanotubes provide high surface area and excellent conductivity, facilitating efficient electron transfer and enabling the development of highly sensitive sensors. Graphene, with its two-dimensional structure and exceptional conductivity, supports high loading of biomolecules and enhances signal transduction. These materials can be functionalized to improve selectivity and stability, making them ideal for advanced sensing applications. Metal nanoparticles, especially gold nanoparticles, are widely used in electrochemical sensing due to their catalytic activity, biocompatibility, and ease of functionalization. Recent trends emphasize environmentally friendly synthesis methods, including plant-based approaches that reduce the use of hazardous chemicals. Gold nanoparticle modified electrodes exhibit enhanced sensitivity and stability and are commonly employed in immunosensors and biosensors for detecting biomolecules such as hormones and proteins. The integration of nanocomposites, materials composed of multiple nanoscale components, has further improved sensor performance. These systems combine the advantageous properties of different materials, such as conductivity, catalytic activity, and biocompatibility. Nanocomposites facilitate biomolecular mobilization, enhance electron transfer rates, and improve overall analytical performance. Future perspective and conclusion. Despite significant advancements, challenges remain in the development of electrochemical sensors, particularly in achieving high selectivity in complex sample matrices and preventing interference from non-target species. Future research is expected to focus on improving recognition elements, developing multifunctional sensor arrays, and integrating sensors with microelectronic systems for enhanced data processing and connectivity. The emergence of wearable and implantable sensors, along with advancements in nanotechnology and materials science, is expected to drive further innovation. In conclusion, electrochemical sensors represent a versatile and powerful class of analytical tools with wide-ranging applications across scientific and industrial domains. Their inherent advantages, including high sensitivity, rapid response, and adaptability, make them indispensable for modern analytical chemistry. Continued progress in materials science, nanotechnology, and device engineering is expected to further enhance their capabilities, enabling more efficient, accurate, and accessible sensing technologies for future applications.